Welcome to another edition of CCSD Living Environment. Today we're going to finish talking about cell growth, division, and reproduction. And to do that, we kind of need to look at part 2 of the process of cell division. So we started talking about the chromosomes in prokaryotic and eukaryotic cells. And what we're going to do now is we're just primarily going to look at and talk about eukaryotic cells and what happens. The cell cycle, as a cell grows, it prepares for division and it divides to form 2 daughter cells. So remember, when that cell splits in half, each new cell is called a daughter cell. Each daughter cell moves into a new cycle of activity, growth, and division. And this just continues on for as long as those cells continue to thrive and reproduce. The eukaryotic cell cycle has 4 phases: G1, S, G2, and M. The length of each part of the cell cycle and the entire length of the cycle changes depending on the type of the cell that you're in. The first phase that we're going to look at is called interphase. Interphase is the time between cell divisions. So from when the cell stops dividing to when it starts dividing again, that is called interphase. And interphase is divided into 3 parts: G1, S, and G2. So we said there were 4 phases. The first 3 phases are all falling under what we call interphase. So let's take a look at what's actually happening during those steps. During the G1 phase, the cell is growing. It's doing its normal job. So the cell will increase in size and make new proteins and organelles. So our cells most of the time happen to be in interphase, and they're usually undergoing G1. They're doing whatever it is that they do. That then moves them to the S phase, or the synthesis phase. The DNA replication takes place during that phase. The new DNA is made when the chromosomes are replicated, and the cell has twice as much DNA as it started with. We don't want twice as much DNA as we're supposed to have, but we somehow have to make sure that we're going to have equal amounts of DNA in both of our new cells. So the cell will undergo S phase, or synthesis phase, to synthesize the DNA or to replicate the DNA. Then we move to G2. The G2 phase is simply when the cell is preparing for cell division. The shortest phase, mind you, and many organelles and molecules are produced. You have to make sure that when you split to give you 2 new cells, that both of those cells are going to have equal amounts of what they need for that cell to continue. So the G2 phase is just getting the cell ready to actually undergo that cell division process. And so finally that takes us to the last part. So now we're stepping out of interphase. Remember, interphase is just G1, S, and G2. That moves us then to the M phase, which is actually cell division. This is where we produce the 2 daughter cells. This phase is named after the process of mitosis. So mitosis gives us the M phase. So mitosis starts another phase, because remember, interphase was G1, S, and G2. So the M phase, we're out of interphase now. So what are we going to do? There are 2 major stages to cell division: the division of the nucleus, which is actually called mitosis, and the division of the cytoplasm, which is called cytokinesis. Remember, cyto means cell and kinesis means to move. So we're moving those cells, if you will look at it that way. So what I've done is I've put the cell cycle up here for you to take a look at. The picture's also in your note packet. I go G1, S, and G2. All of those belong to which phase? Yep, that's right, interphase. And then I go to the M phase, which is actually what? Mitosis. And what did it tell us mitosis is? The division of the nucleus. So what we need to do is we actually need to work our way through the steps or the phases of mitosis. So remember, interphase starts us off. The cell is doing its job, then the DNA replicates, Once the DNA is replicated, now the cell can get ready to actually undergo mitosis or cell division. So what are the stages, what are the phases of mitosis? The first one is called prophase. There's a couple things for you to notice. These little things here, which we'll come back to in a minute, notice that that circle that's usually in the middle to represent the nuclear membrane is starting to form a dashed line, and you can actually see the chromosomes inside the cell. And what kind of chromosomes are they? They're double-stranded chromosomes. Why? Because the DNA replicated during interphase. So in prophase, it is the first phase of mitosis. It is usually the longest phase. The replicated chromosomes have condensed and become visible. I can see them. Outside the nucleus, a spindle, which is what these guys are, a spindle starts to form. The nucleolus and nuclear membrane disappear. And that's what the significance of that dashed line instead of the solid line for the nuclear membrane is representing to you. So the nuclear membrane is actually going away. After prophase, we move to what they call metaphase. So again, here's my spindle. It's got something attached, and you can see my chromosomes. And there's a very important thing for you to notice during metaphase. The chromosomes have moved to the middle. So metaphase, they've gone to the middle. Metaphase is the second phase of mitosis. It is usually the shortest phase. The centromeres line up in the middle of the cell. What was the centromere? If you think back to that picture we had of single and double-stranded chromosomes, the centromere was the center part of the chromosome. So what we're trying to show here is that it's the center that's attached. So the centromere is how it's attached. Spindle fibers connect the centromere to the spindle. So remember, this structure was called the spindle. This is the spindle fiber, and the centromere is attached to the spindle fiber, which is then attached to the spindle. Okay? So our next phase then is anaphase. So in anaphase, you can see our spindle, you can see the spindle fibers, but what do you notice about the chromosomes? That double-stranded chromosome that I had has separated, and they're starting to move away. So anaphase, away, A away. During anaphase, the third phase of mitosis, the sister chromatids, which is what these guys were when they were together, the sister chromatids are separating and moving apart. They begin to move to opposite poles along the spindle fiber. So if you think about the spindle fiber like a zip line, and think about the centromere as the harness that that chromosome would be wearing, it's sliding along that spindle fiber. That's how it can move from one side of the cell to the other. So they separate from each other, and then they can slide away to the opposite poles, or to the opposite sides of the cell. So then what? Now look in the next one. The spindle doesn't seem to be very important, but what do you notice here? You can see where that nuclear membrane is starting to come back, and what can you tell me about my chromosomes here versus my chromosomes up here? Yeah, that's right. These are single-stranded chromosomes. That was the whole idea. We replicated the DNA to give us a double-stranded chromosome. They separated, so now each new cell is going to have one copy of each of those chromosomes. So during telophase, which is the fourth phase of mitosis, the chromosomes begin to spread out and are no longer visible. The nuclear membrane starts to reform in both cells. So the nuclear membrane starts to come back, and the nucleolus will come back as well. So that takes us through the stages of mitosis, the nuclear division. How did the nucleus divide? What's the other thing that has to happen? The cell itself has to divide. So that process is called cytokinesis. It usually occurs with telophase. So we're representing it like it's happening at different times, but generally speaking, when telophase occurs and the nucleus is divided, the cytoplasm will continue to give you 2 new cells. So with cytokinesis, it completes the cell division. It splits one cell in 2, and the actual cytoplasm is split in half. We need to split that all the way so we have 2 new individual cells that are identical to each other and the original cell. Absolutely. So when we go through this process, you can see that I have the 2 nuclei formed, and notice I'm back to not really chromosomes. So what are we going to call that? It's that tangled ball of DNA. And that was the chromatin that we talked about in the end of the last video. So when we start the whole process, the cell has chromatin. The chromosomes then become visible. And when we end the process, we're back to chromatin. It's all wrapped up. The DNA is wrapped around those proteins called histones. And that gives us chromatin. Okay. So let's talk about plant and animal cells. We know that there's differences between plant and animal cells as far as their structure, but there's also a difference when it comes to how they separate their cytoplasm. So plant and animal cells divide their cytoplasm differently. And I've tried to kind of show you a picture here. In the animal cell, you can see my nuclear membranes have started to reform, and you can see that the chromosomes are back to that chromatin. With my plant cell, well, it's not quite doing the same thing. Notice this line in the middle. But the nuclei have reformed, and we have the chromatin in the middle. So there's some similarities there. The difference is in what happens next. With an animal cell, remember there's room for animal cells to kind of move around. So those cells can split in half and go their separate ways. With a plant cell, remember looking at plant cells under the microscope, it's almost like a brick wall. There's no place for them to go. So when that plant cell is ready to divide, what happens is a different structure forms. So we're going to hold on to that for a second and talk about the animal cell first. The animal cell creates this structure called a cleavage furrow. Furrow meaning where it starts to pinch. So it's formed when the cell membrane literally pinches in. The cell membrane is drawn in until the cytoplasm is pinched into almost equal parts. Each part has its own nucleus, and each part has its own cytoplasmic organelles. So when this pinches all the way, each one will have its own organelles, it has its own cytoplasm, has its own nucleus, has its own DNA. They're 2 individual cells that can continue then on their own. So what are we going to call this structure in the plant cell? That structure in the plant cell is called a cell plate. The cell plate forms halfway between the divided nuclei in a plant cell. Why does it do that? The cell membrane isn't flexible enough to pinch in because of that very rigid cell wall in the plant cell. Remember those cell walls that provide that extra support and structure for plants? Well, it doesn't allow the cell membrane to actually pinch. So what happens, the cell plate will eventually become a cell wall for the plant cell. But in that beginning part, when it's starting, oh, okay, I can't do this, so what am I going to do? The structure forms between where the 2 new cells will be, and eventually it creates a new cell plate, so now you have 2 individual plant cells. So this video actually took us through the process of cell division and how cells actually undergo cell division and what they do to get there. So hopefully you can remember interphase, prophase, metaphase, anaphase, telophase, and remember during telophase, cytokinesis takes place. That wraps up the video for today. Have a great day.